Look closely at this microscopic footage. These neural connections are actively dimming and detaching from one another, the decay of adult neuroplasticity in real time. This scaling down of the brain's infrastructure translates directly into the symptoms we often mistake for standard aging: profound mental fatigue, a reduction in cognitive stamina, and difficulty consolidating new memories. To maintain these connections, the brain synthesizes brain-derived neurotrophic factor, or BDNF. This protein acts as an internal maintenance crew, keeping existing neurons healthy while building the synaptic bridges required for new learning. Measurable cognitive decline tracks closely with the natural drop in these BDNF maintenance protein levels. Upregulating BDNF presents a significant biological challenge. While it is required for memory preservation, production naturally falls with age, and the blood-brain barrier prevents simple pharmaceutical replacement through pills or injections. A non-invasive alternative exists in the physics of sound. Specific acoustic frequencies can intercept neural processing to stimulate the genetic expression of BDNF. Tracing this pathway requires looking at how an auditory signal moves from the cortex down to the microscopic architecture of chromosome 11. Before we break down the frequency math, please like this video, subscribe to the channel, and hit the bell icon. You can find a link in the description to explore these findings further. Restoring the brain's maintenance system depends on understanding how acoustic waves interface with our biology. To understand the trigger, we first look at the target. BDNF neurotrophic factor is found in its highest concentrations in the hippocampus, the brain's primary center for learning and spatial memory. Within the hippocampus, BDNF facilitates long-term potentiation. This is the process where temporary electrical activity is encoded into physical stored memory. The mechanical interaction is precise. The BDNF protein binds to a TKB receptor on a dendritic spine, triggering a molecular cascade that causes the synaptic connection to physically thicken. Permanent memory storage requires this physical structural growth, which depends strictly on the availability of BDNF. The protein itself originates from the BDNF gene located on chromosome 11. While genetic variations like the Val66 MET polymorphism influence an individual's starting point, baseline BDNF production is responsive to environmental inputs. The architecture of this gene is complex, featuring 11 exons and 9 distinct promoters. This structure allows the gene to ramp up production in response to specific external signals. Activating these promoters serves as the biological switch for cognitive repair. Environmental activation depends on the brain's electrical state. Our neural activity oscillates across a spectrum of frequencies, ranging from deep sleep to high-level arousal. The target for peak cognitive performance is the gamma state, which oscillates between 30 and 100 Hz. Research has identified a correlation between sustained 40 Hz gamma activity and measurable increases in BDNF levels. Inducing this state requires psychoacoustic stimulation delivered through stereo headphones. 40 Hz gamma waves provide the electrical environment that activates the BDNF gene's promoters. Isolated stereo audio creates a unique neural computation. When each ear receives a different frequency, the brain must reconcile the difference. Here, the left ear receives a 240 Hz wave. Simultaneously, the right ear receives a 280 Hz wave. The brain resolves this discrepancy by generating an internal 40 Hz beat. This signal propagates, entraining the cerebral cortex into a gamma state. If you're interested in the data behind this entrainment, please like, subscribe, and check the link in the description for more. This auditory task effectively shifts the brain into its highest performance state. This three-panel view maps the entire biological journey. The acoustic differential creates gamma entrainment in the cortex. This electrical state acts as the environmental trigger for the promoters on the BDNF gene. Genetic transcription increases BDNF production, allowing for the physical fortification of synapses. These stages illustrate a pathway where specific acoustic stimuli influence both cellular structure and genetic expression. Applying these findings requires a daily 17-minute protocol of binaural listening. This duration is based on psychoacoustic research, indicating that sessions averaging 20 minutes provide the optimal window for memory enhancement. The 17-minute calibration provides the sustained signaling required to initiate structural changes. This non-invasive approach works with existing biology, providing the conditions the BDNF gene needs to execute its natural repair functions. To learn more about the specific audio protocols used in this research, see the link in the description. Like, share, and subscribe to stay updated on new developments in neuroplasticity. Supporting a high-functioning brain is a matter of providing the nervous system with the frequencies required for its own maintenance.